Great, thank you so much, Duncan. We are going to, we have 15 minutes now for a question and answer. If you want to raise your hand or add it in the Q&A. Going to have you stop sharing, Duncan.
Mm-hmm.
And then attendees, feel free to raise your hand, add a question in the chat, and we'll be watching both. We do, yes, thank you. Okay, we do have a question here in the room from John Woodward. Go ahead, John.
Question for Duncan. How was the motion added to the 3D models?
Right, so we actually have developed a machine learning algorithm that can take motion from an animal and put it into a model. We published that 2 years ago, and I'm happy to send that paper to anyone who's interested. But for these particular models, that was honestly just for fun. We just added the motion in based on our knowledge of fish, shark locomotion. So we actually create both static models and also we create just animated models, and the animated ones primarily get used for education, but the static models are the ones that get used for sort of research and applications and things like that.
I have a couple of, does anybody else in the room?
Beth Brainerd.
Yeah.
Yes, but there's a couple questions.
Yeah, there's a couple of follow-up questions to that one. So Alana Rivera asks, how much of your work relies on a still or relaxed individual?
Yeah, a lot of it, yeah, absolutely. Like in the case, but not always. Like in the case of the hammerhead shark, they were never still. I think we were filming at video. I think we were filming at 120 frames a second. So in the case of, you actually can use video as long as the quality is sufficient of the video to recreate motion capture, and that's been well documented in literature. But obviously a still animal like a frog or a reptile that just remains rock solid still is ideal. It's a little technically easier for most people, but we can do both, and we have done both, and others have done both as well.
And then another question is, can this be used to create synthetic data?
Yes, so the digital model is one of the biggest areas of growth that we've seen from other partners. Is now that we have, for example, 3D rhino, there's a lot of discussion of, it's very hard to scan rhinos, as you can imagine, but there's the idea that to use 2D images of rhino that are very common, to actually use that machine learning algorithms to then create lots of digital copies of rhinos from our initial 3D model. And there's a huge growth of industry of 2D to 3D. So I think there's a lot of thought about using our 3D rhino to scale and calibrate and create new synthetic data, which makes sense because obviously scanning rhinos around the world is not a very practical enterprise. So yeah, I think absolutely that's a big growth area.
Any other questions? Yeah.
Another question. So also this is open to all the talks, these questions. So questions for the other discuss them. But Duncan, I was interested. So you have a lot of movement of the animal that's walking around. How does that affect the alignment when you are doing your initial alignment in the reconstruction program?
Yeah, so that's a great question. So that's actually where our team, we actually have a lot of students that increasingly are coming to our lab to learn these techniques, but a lot of the ideas about photogrammetry is with commercial software like Agisoft. But actually what we've been primarily focusing on is developing using Blender to reconstruct models. So for example, you don't really need the 3D photogrammetry software to reconstruct a shape. You can actually bring in those images into Blender and actually, even if an animal's moving, can align those photos manually. It's just a more involved technical process. So yeah, the fact that I guess in short, what we've been doing is not going to be so easy for, let's say, a high school student who wanted to learn photogrammetry. They would just more scan a rock and use Agisoft or whatever. But if you have, let's say, a shark that's swimming or a fish that's swimming, you can absolutely create a 3D model. You just have to use other software. And I didn't have time to go into it, and I'm happy to send some publications out that lay out this foundation. We do, and I should say for any of you that want to learn this process, which is not well known, by the way, a lot of, I have a student this who's here at my lab for a week. We have a lot of students that come through my lab and we just teach them how to do this. And usually after about a week, they figure it out. So it's a skill set we're trying to share with the community.
Michael.
Yeah, I'm interested in, this actually might be for all three speakers, but particularly for Nelson and Duncan. Both of you mentioned capturing color. These different techniques, and which is something that does not preserve well for many tags in traditional specimens. So I'm wondering, what's the prospect for doing that? How well developed is that? Do you need to use calibrated cameras? Just those sorts of things.
Yeah, I could address what we do. So, I mean, in our mounts, when we're doing 3D imaging, even when we're doing, less dense image sets, we always put a little color checker in there, a standard color checker, so that we can calibrate. We do most of our shooting in RAW, and then we can do the color correction afterwards. It is an extra step in the process to do that. But yeah, that's what we do.
Yeah, for our, I didn't have time to talk about it. My graduate student, Josh Medina, has been developing a way to quantify color analytically from photogrammetry data. It's quite an involved process. He's been pioneering with birds. And the key thing is it's fairly straightforward just to get color data from photogrammetry, but it's a whole nother thing to develop a kind of a whole analytical package around it. So that's what we've been working on, but we haven't published that data yet. And he'll have a paper he'll put into review in the next month or so that'll address that. I'm very excited about it because I think it's actually allows you to map on color data onto shape data pretty accurately. I think it'd be for collections, it's going to be fantastic. So.
And we have, oh, sorry.
No, Nicole, did you want to add anything? I feel like
Yeah.
we're dominating the conversation here.
Oh, no, it's totally fine. I'm not using currently color checker data right now in terms of in the field surveys of using 360, particularly because I think it'll be more difficult as well to find color checker. I've actually found color checker data with the flattened out images to be a little bit more difficult to apply. So that is something we're also working on too.
And also for you, Nicole, have you played with underbrush or forest floor for omnidirectional work? The person who's asking the question is curious if there's any sort of environmental density challenge. For example, if there were branches in front of a cardinal.
I have not played with underbrush or forest floor for the omnidirectional work. We've been focusing largely on capturing actually a large portion of a field site too, and working to see what ground pattern that we can get perhaps the most accurate representation or something that's a little bit closer to accurate. And I think I can see there was another answer maybe or a question about accuracy as well. But yeah, we have been largely trying to see if we can capture the multiple sizing of specimens in a space. And there's been really interesting interaction between the density challenges as well. If you noticed in my slide, I did have 2 layers of the 360 camera. So I had it at about this high, and then I used an extension that reached it higher, which makes up for some of that overlap. But there's still kind of those struggles with overlap and environmental density too.
Thank you.
So there's been some interesting work done in Cork, Ireland. I cannot remember the researcher's name, she's a paleontologist, and she has tried to reconstruct color from fossils. Yes, right, and pretty fascinating and more challenging than trying to reconstruct from, I think, even specimens. I don't know how she's doing that. Also, back to John's question regarding movement and how movement that was shown on the leatherback. Beth Brainerd at Brown has done a lot of work with birds and trying to understand the internal anatomy and how birds function through adding to specimens movement from bird images. Austin.
I have a question for David. So I really like the talk and I like the board. So it's such a big, diverse audience that you're maybe not even having in mind at the time that you're creating the models, but instead just creating the models and seeing who will come. Am I wrong in that? Is it the case that you've identified from the start these 5 groups that are personified by the artist from here and the 5th grader from there, and how do you think about your audience?
Yeah, I think it's actually a really interesting question. I don't think it's something that we went into it thinking hard about, other than just thinking about scientists. Scientists want these data somehow and they'll be useful somewhere else. But yeah, I think it's partly from the fact that I've approved so many thousands of download requests that, those all come from people with names and they write about what they're going to do it. So I think that's part of what's kind of made me think hard about it. And Ed and I have talked a lot about, using the download information we get to think about, like, well, what is it people really are gravitating? Like as systematists, many of us are in the museum community, we tend to think type specimens. But it's like, okay, well, I don't think type specimens are really that useful to people outside of systematics, right? Usually, like, they're the worst ones. But I do think that, like I'll just give an example. One of our most used specimens from over was a WIM. It's not even a fluid preserved specimen. It was a skull of a gharial crocodile that one of my grad students decided to just put in the scanner and put online. And it gets used constantly for science and outreach around. And that just has made me think, when we talk to a lot of collection folks to pick out specimens, there's a lot of specimens that would meet our criteria. Like, all we need is one individual of a species of a genus. Like, we have plenty of individuals, of an anole. Like, which one are you going to pick? The ones that we would often try to guide people to is ones that might have some other story of interest outside the research. Like the last one ever seen in place X. I used to joke that if you got a choice between picking a specimen collected by Dave Blackburn or Charles Darwin, like, you'll always pick Darwin, right? Because there's going to be school teachers that are suddenly interested in it, right? Because of that. So I think there are audiences, we tend to have this, maybe sometimes it's explicit, most of the time it's implicit, that we think that somehow the work that we do related to digitization kind of connects to DEI issues and accessibility. More people can now use the stuff behind the scenes of the museum. But our sort of brief foray into looking into that based on user data doesn't suggest that's true. It's basically just more of the same people we already interact with, which is fine. But at the same time, it kind of speaks to where if we're really the goal is to reach outside, that community with these data, then we need to be a little purposeful. And I think going through usage statistic data can get you some sense of like who your users are and how you might make things different. Ed or Jamie, we've talked so much about this. You have thoughts?
No, I mean, I'm going to talk a little bit about that, but I mean, the issue I think is we need people to have the tools to work with the data that we're putting out there, right? And I say we're constantly modifying the setup of the data that we're presenting to people as the audience changes. That's the kind of dynamic aspect that I think we really need to think about. As you say, start with the scientists, but as more and more people find out about this kind of digitization work, the disconnect between what we're providing and the audience, what the audience needs, grows, I think. So we need to plan around that.
Just to not drag this on, but one comment of that is one specific way in which we've responded to people is that for those of you who interact with Ogre, you'll know that at least at UF, it was possible to jump the scanning queue because if people asked that they needed this specimen or these data for some specific project, we usually just bump that up in the queue. Maybe it wasn't something we were even planning on doing, but I was like, if the whole point is to have people use it, what better way than to have people ask for it and then give it to them, right? And so I think, that kind of over the course of Ogre, we kind of evolved in that to where we were in fact sometimes responding to requests.
Other good questions? So far, we've had a lot of good questions.
This time, I was real quick question about the 360 videos. I was wondering if anyone's played around with stereoscopic 360 videos. So multi-camera from multi different 360 with multi cameras that provide stereoscopic. Okay.
We track firefly swarm movements with 360 stereoscopic to understand collective behavior. It works really well to make 3D trajectories of these behaviors.
That gives you additional pinpoint accuracy to those things. That's great.
Yeah.
What system did you use?
Like what cameras?
Which camera? Yes.
Yeah, we just use GoPros, and
Okay.
we have citizen scientists that will send GoPros to also to collect data for us.
Okay.
And it works really well. It's great at tracking movement data, and because fireflies flash on and off, we can do it at night. But you do need light typically, but you can do it with like birds or other things too.
And use two.
Yeah, so you either put them 3 meters apart or 6 meters apart. We have people tell us how far apart they are. We put them in a, I didn't make the algorithm, but one of the people in my lab made an algorithm, and it'll spit out XYZ coordinates and make animations of the movement.
Thank you.
Nelson, you talked about focus stacking. It seemed, and I may be wrong about this, but does your compass system sort of make focus stacking superfluous? I mean, you're taking images from all kinds of directions, so you must be getting some inherent focus stacking out of your method.
Well, so we still have to do focus stacking when we're limited by the optics that we have, right? So, and it all depends on the environment. I mean, what's really nice is since it's so controlled, and we could, in essence, leave the shutter wide open for 10 minutes and have a really long exposure of gaps. And so that could give us a really deep depth of field. But we try to do something that's more reasonable. And sometimes for insects, we do need focus stacking to get that high resolution.
Yeah, right.
So with the optics that we have, usually if we're shooting something that's smaller than 7 or 8 millimeters, we probably have focus stack.
Okay, we are at time. Next is going to be Zach Randall from the Florida Museum. Okay.